Hi everyone, my name is Ting-Lun Chang, and today I'm going to be talking about my spring Europe research topic, which is neural correlates a voluntary movement in the human basal ganglia via extracted SEEG single neuron data. Now, let's jump right into it. What is the basal ganglia and what is dystonia? Well, the basal ganglia are a group of subcortical nuclei involved in motor control, learning, and habit formation. Now, basal ganglia dysfunction is actually something that's well studied in movement disorders like Parkinson's disease, where we're able to use that knowledge to have informed deep brain stimulations to mitigate the effects of this disorder. However, direct correlations between basal ganglia neuronal activity and dystonia is something that is still remaining to be largely unexplored. Dystonia is a neurological disorder that is characterized similarly, but in some ways, to Parkinson's disease with involuntary muscle contractions and abnormal postures. Currently, in Dr. Sanger's lab, in collaboration with the Children's Hospital of Orange County, my research focuses on investigating basal ganglia neuronal activity in pediatric dystonia patients undergoing deep brain stimulation surgery. Utilizing stereoelectroencephalography or SEEG electrodes, we are able to capture high resolution spiking activities directly from targeted regions within the basal ganglia. These recordings are attained during controlled voluntary reaching task, which offers unique insights into neuronal processes underlying motor dysfunctions in dystonia. Now, what do we do with all this data once we have the recording? Well, in our studies, we have developed a novel spike sorting algorithm that is specifically developed for the SEEG microelectrodes used in our study, as well as commonly utilized in DBS procedures. Now, our methodology includes using advanced signal processing techniques that are used for noise and artifact removal, as well as adaptive thresholding and also nonlinear filtering in order to robustly detect neuronal spiking activities. And then also subsequently, we use wavelet decomposition in order to have feature extraction for our spikes. And then lastly, finishing up with Gaussian mixture model for clustering. And doing so, our refined spike sorting approach is able to reliably isolate the genuine spiking activities from neuronal signals from the complex SEEG data. But why is any of this important? How does this affect treatment? Well, first of all, we're working to clarify how basal ganglia contributes to voluntary motor functions, especially in pediatric patients where this circuitry remains largely unexplored. By analyzing the brain activity during movement, we hope to uncover the specific neural mechanism underlying motor dysfunctions in dystonia. These insights can help guide us to have development of more precise and effective deep brain stimulation treatment for our patients, and ultimately using our work to support the creation of a closed-loop stimulation for neuromodulation system where the technology can detect abnormal brain activities in real time and automatically adjust stimulation to improve outcomes. Thank you so much.